This short video is going to be talking about the second part of translation, which is the converting of the mRNA that was produced in transcription to the actual sequence of amino acids that make the protein at the ribosome. What do we need for translation? Well, we need mRNA because that's the message. We need rRNA because that's the ribosome where things are being built. And then we need the tRNA because that's what's carrying the amino acids to the ribosome. Talking about tRNA in more detail, since it has the job of collecting the amino acids that are floating around in the cytoplasm, it has to know which one it's supposed to pick up. The amino acids are going to be seen here as the little circular blob. The tRNA is a single strand of RNA that is folded on itself to kind of look like a T. And then at the bottom, opposite the amino acid, there's a group of 3 nucleotides that is significant, which is called the anticodon. Remember, I told you that there were names for groups of 3 nucleotides, depending on which nucleic acid we were talking about. A group of 3 on DNA, remember, was called a triplet. A group of 3 nucleotides on mRNA was called a codon, and now a group of 3 nucleotides on tRNA is called an anticodon. If you're having trouble remembering which one goes where, I like to think of there's a T in anticodon, and that is for tRNA, if that helps. Steps of translation. mRNA comes out of the nucleus after it was edited. Remember what were the garbage pieces that were taken out? Those are the introns, and then what were the parts that were glued together to make a continuous message? Those are the exons. The ribosome attaches to the mRNA, and the anticodons of the tRNA are attracted to the codons of the mRNA, and they follow the same rules as before. If a codon said AUG, then the anticodon would read UAC. Can we use T's in the RNA world? No, we can't. And so that's why it would be this sort of pairing rule. When 2 tRNAs are next to each other, there are 2 amino acids next to each other. And so those 2 amino acids then connect with a peptide bond. As we continue to form peptide bonds, we then have a chain of amino acids. When we get to a stop codon, the ribosome falls off, and we're done. The product is going to be a chain of amino acids, which we can also call a polypeptide chain, which is a protein. Poly means many. Peptide refers to the bonds that are being formed between our amino acids, and then you got your chain. So we go back to this image, which is the last side of the last page, and we see the translation is happening out here. Here is the, where's my pen? Come back. Oh, there you are. Here's your mRNA over here, adding to the ribosome. Remember, the ribosome is made of rRNA. And as the mRNA comes through, the tRNAs are going to come together and link these amino acids together.
Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene.
Remember, this is your DNA.
It moves along the DNA,
It's
making
up.
a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA.
Okay, that was an intron that was taken out. Exons are what?
The messenger
are glued
RNA
together.
then moves out of the nucleus into
See
the
those little holes?
cytoplasm.
DNA's too fat, can't come out.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids.
There's your
There are
anticodon.
20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time.
Remember those 3 bases were called a codon, and then you got your anticodon, sorry that's blurry, that is going to use the base pairing rule so they know the order that the tRNAs are supposed to line up.
As each triplet is read, a transfer RNA delivers the corresponding amino acid.
As these amino acids are linked together, these linkages are called the peptide bonds.
This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
There you go. That's a nice visual. You're welcome. Okay. First thing, you need to find the page on your notes, and hopefully you can see it, where you've got what's called the circular genetic code that I've got here on my screen. And then you've got another set of concentric circles next to it. And then you've got my mRNA message at the bottom. So find this page, please. First thing I want you to do is, if this is my mRNA, I want you to divide it into codons. Remember, codons are a group of 3 nucleotides. How many codons is this message? Does this message contain? And I'm going to do little vertical lines that separate them. And so if we count how many codons there are, 1, 2, 3, 4, 5, 6, 7, 8 codons long. The messenger RNA is going to code for the amino acids that we have. Well, how are we going to read that? Well, if we go to this page where my message is gone, I want you to look at the concentric circles. This middle, or this most central circle, is going to be the first nucleotide of the codon. And then you look at the second nucleotide, and that's going to be within the second ring. So for example, our first codon was AUG. So A is here, and then we've narrowed it down to this quarter of the genetic code. The second nucleotide was a U. So now we've narrowed it down to this sector. And then this next ring out is going to be the third nucleotide of the codon. And there's G is the third one. And so what amino acid does AUG code for? It codes for one called methionine. I would like you please to use these abbreviations in this ring, not the singular letters that are out here. Just use the MET for right now, okay? So this is going to be your amino acid, this ring, and this is the code that we're going to be using. Now, just so that you are, well, no, I'll show you that later. Here are the codons. These would be the anticodons of the tRNA that would be carrying the amino acid, but I want you to make sure that you are looking at the mRNA, because the mRNA is what codes for the amino acids, okay? These are the ones that are just going to carry it. So go ahead and fill in your anticodons. A goes to U, U goes to A, G goes to C. That's your first anticodon, and then U, A, G, and so on. But what are the amino acids? We know that the AUG is going to give us methionine. Then what's AUC? AUC is isoleucine, which is an ILE, isoleucine. GUA, GUA gives you valine. And then CAA, CAA gives you glutamine. UGC, UGC gives you cysteine. GCU, GCU gives you alanine. And then UCC, UCC gives you serine. And then UAA is a stop. Is that an amino acid? No, that just means the end. So I'm going to put stop here, meaning that I know that my amino acid chain is really only 7 amino acids long, because that was the end. I like the circular genetic code, and this is what I'm going to provide for you for an assessment, because you're going to have to be able to produce for me the amino acid sequence if I give you the mRNA code. But you might find on a later assessment that you're going to have it in a chart form. And it's the same sort of idea, is that the first base is here. So if we were to look at AUG, we would say, okay, A is the first one. So that narrows it down to this row. U is this column. So now we've limited it to this chunk of 4. And then G is over here. So that's going to give me this one here, methionine. This is the second way to read the code. You are going to have to, number one, if I give you an mRNA code, you will need to be able to break it into codons, and you will need to be able to tell me the series of amino acids that is coded from that mRNA. But there's also going to be the reverse. The reverse is if I say, hey, what are the codons that would produce a proline? Well, you find proline on the chart, and you say, well, what are the different ways to get a proline? Well, it looks like there's 4 ways to get a proline. And so I would write from the center, I would say for proline, I would say CCU, because, here's C is the first one, C is the second one, and then U. Or I can have CCC, or I can have CCA, or I can have CCG. 4 different ways to get a proline. What about leucine? Leucine is over here, so I would have CUU, CUC, CUA, CUG, but, there's also a leucine over here. So there's actually 6 ways to be a leucine. So you need to be able to go in both directions. If I give you the codon, tell me what the amino acid is, and then if I give you the amino acid, tell me what possible codons would code for it. And then obviously, if I give you a codon, you need to be able to tell me the anticodon that would link with it when the tRNA is bringing that amino acid there. Okay? And quite frankly, you also need to be able to go from triplet from our DNA to codon to anticodon and vice versa. Okay, that'll do it for this installment.